Hello everyone. We have previously discussed that antibiotics are used to fight infections caused by bacteria. Unfortunately, sometimes a major problem can occur, and it is that some kinds of bacteria develop resistance toward the antibiotics or drugs, which means that they are no longer killed by this antibiotic. So, in this tutorial, guys, we will learn how do many types of bacteria develop resistance to a certain drug. We will also learn how do doctors perform the test to know that certain bacteria are resistant. It is interesting to know that germs such as bacteria will always look for ways to survive and resist new drugs. That is why they use certain mechanisms in order to fight the drug and develop resistance. For example, they might modify the cell wall protein and make it impermeable, which means that doesn't allow the antibiotic to enter. Moreover, they can modify the shape of the antibiotic. So, when the antibiotic changes its shape, it will no longer have the same effect. Moreover, they can create pumps which forces the antibiotic outside the bacterial cell. Finally, they can inactivate the antibiotic by adding a phosphate group to it. Once they tag it with a phosphate group, the antibiotic will no longer be able to bind to the ribosome. Consequently, when these mechanisms are followed by the bacteria, resistance will be developed. And so, doctors will have to develop new drugs in order to kill these bacteria. However, how do doctors know that a certain strain of bacteria is resistant to a certain type of drug? In order to know, doctors perform the susceptibility test. What happens in this test is that bacteria are grown on an agar plate. Then, different types of antibiotics are distributed to the plate and left for 24 to 48 hours. Then, what doctors do is that they observe the inhibition zones. As you guys can see, the wider or the larger inhibition zones means that this antibiotic is very effective in killing this strain of bacteria. The places where no inhibition zone is observed near the drug means that this bacteria is resistant to this drug. That is why the drug couldn't kill this bacteria and create an inhibition zone. Inhibition zone, by the way, means that there's no growth of bacteria. So, the places with no inhibition zones means that this type or this strain of bacteria is resistant to this drug.